one other issue I want to discuss, which is not directly related to genetic technology or DNA recombination, but which is, I think, a serious issue, is that of stem cell and stem cell research. Stem cells are cells that are relatively undifferentiated or unspecialized. And when we look at stem cells and how they behave, again, most people are familiar with what we call embryonic stem cells. And embryonic stem cells are kind of like the Ferrari, if you will, of stem cells. They're what we call pluripotent. And what that term means is that embryonic stem cells can really differentiate into any tissue. And again, these would be the cells that we have shortly after fertilization and early in the cleavage stages of our embryonic development. So these potentially and ultimately will continue to divide and develop into all of the tissues of our body. So again, embryonic stem cells will eventually develop into what we refer to as differentiated cells or specialized cells. So skin cells, brain cells, liver cells, kidney cells, whatever they happen to be. Now also, stem cells, embryonic stem cells, will give rise to other stem cells that are not pluripotent, that are more restricted. So within our muscle tissue, within our bone tissue especially, we have a number of stem cells. So I think a lot of people think that all stem cells are embryonic stem cells, and that's not really the case. We have a number of stem cells scattered throughout our body. But the problem is that these progeny stem cells that we have in and amongst our differentiated tissues are not pluripotent and cannot develop into anything, or at least we don't really know how to do that yet. So one of the issues that we've always been curious about is it possible then to take these limited stem cells to a pluripotent state? And also, would it be possible, or is it possible to take completely differentiated cells back to a pluripotent state? And so this is one of the questions that was resolved to some degree when we look at the cloning of Dolly, because there they actually took an adult cell from the udder, merged it with an ovum to produce a cell that really is not technically an ovum and isn't technically an embryo, if you will, once it starts to divide, because it's not the product of sperm and egg. But ultimately, that embryo, if you will, eventually did produce cells that could completely differentiate and ultimately formed Dolly. So the answer to these questions then is it possible to do this? Yes. But do we ultimately know how to do this yet? And the answer to that is no. So when we look at stem cell research then, again, the best tool that we have to use in looking at treatment for disease and so on is embryonic stem cells. And of course the controversy is that those come from embryos, as opposed to using more differentiated types of stem cells. So when we use stem cells, again, there's a lot of potential applications for stem cells, certainly for treatment of disease, and especially when this disease involves tissue damage. But what I also want to mention here is that this is also where there is some overlap with genetic modification, because again, we could take and can take stem cells, genetically modify them in some ways, and in some cases take stem cells from individuals, genetically modify them in some way to perhaps fix a gene and then reinsert them into individuals. Now, again, that's a promising line of research. Certainly we can replace damaged tissues, potentially again with stem cell research, and there's favorable lines of evidence there and research there. Ideally, down the line, we would be able to take stem cells and actually replace organs. We're certainly a long way from that. And also, as cells age and die, again, we might potentially be able to use stem cells to offset the damage from aging, as well as almost anything you can think of. Again, there's potentially thousands of uses for stem cells. So again, part of the point I want to make, though, at this time, however, is that our best tool for this is embryonic stem cells. We don't really know how to take adult cells and more restricted stem cells and use them in these pluripotent ways. And that's the controversy. So I'm not really going to get into that at this point, but there are limitations on embryonic stem cell research right now, and it really limits our effectiveness to look at these treatment of diseases where we have tissue damage, especially, say, brain damage that we have in things like Parkinson's disease, Alzheimer's, and so on. So until we lift those restrictions, we'll have trouble treating those diseases with stem cell research.